Anyone who has spent a few too many hours at the beach knows the exact feeling. The redness creeps in, the skin radiates heat, and eventually the peeling starts. Textbook biology explains this misery through DNA damage. We are told that ultraviolet B light penetrates our skin and strikes our genetic code. To prevent those mutations from becoming cancerous, the damaged cells begin to self-destruct. But the timing of this explanation is flawed. The process of recognizing DNA damage and signaling from the nucleus is slow. It takes far longer than the few hours it takes for fiery inflammation to erupt across your shoulders after you come inside. A study published in Molecular Cell identifies the actual trigger for this rapid inflammation as damaged RNA strands. The painful redness that appears by evening is a separate defense mechanism, operating independently of the body's long-term cancer prevention protocols. This faster system operates in the cell's cytoplasm. Here, messenger RNA acts as a blueprint. Cellular machines called ribosomes travel along these mRNA strands, reading the genetic code to build the essential proteins your body requires. When UVB light pierces the cell, it physically cross-links the mRNA, creating a rigid roadblock. A traveling ribosome hits this kink and stalls. Ribosomes following close behind have nowhere to go, slamming into the stuck machine to create a microscopic pileup. This specific type of molecular traffic jam triggers a cellular alarm known as the Ribotoxic Stress Response. This structural pileup on the mRNA initiates the body's inflammatory response, acting as the starting point for a sunburn. The cell recognizes these collisions using a specialized sensor protein called ZAKa, which physically binds to ribosomes once they have crashed. ZAKa then activates two distinct pathways. The first is apoptosis, an organized cellular suicide responsible for the skin peeling that occurs days later. This diagram shows a second pathway, pyroposis, a fiery cell death causing hot, red inflammation. Researchers exposed mice lacking the ZAK gene to UVB light. Wild-type mice developed inflammation, but mice without the ZAK gene, marked with red X's, developed no acute inflammation. With ZAK-alpha removed, the immediate symptoms of a sunburn never appear, establishing this protein as the critical sensor for sun-induced inflammation. DNA mutations remain the primary long-term threat of sun exposure, but we now know the immediate agony of sunburn is driven by the ribotoxic stress response. Identifying ZAK-alpha as the trigger provides a clear target for new treatments. Scientists can now work toward developing drugs to block this pathway, potentially treating severe sunburns and chronic inflammatory skin diseases. Under a microscope, a sunburn is the cumulative result of billions of molecular traffic jams occurring across the skin. The resulting heat and redness represents an ancient ribosomal alarm system executing a protective protocol to keep the rest of the body safe.